Good morning, everybody. I'm Dr. Bogoman. Students normally call me Dr. B. I welcome you to our first Chuck Lorre Research Scholars Program Science Symposium. It's exciting for me to be here, but I'm not by myself. I have Dr. Manal Tawa. Come on up, please. And she has worked with me for the last 7 months with the program, with the whole intention that once I sail into the sunset, that she's going to take over. Okay, and today is going to be some sort of an exciting day for all of us, not only the students. I wanted to make sure that we recognize the people who helped us to organize the symposium. The person who works with Manal and myself close is Emmanuel, but he currently has fun in New York, so I hope he maybe tunes in. That would be nice. He was instrumental to get this whole thing going, so thanks again. There are people in the tech pad, the education section of the Institute, who are helping us with the little but most important details. I want to thank them also. For the people who currently are online, I'd like to welcome as well. And I hope there will be some of the mentors and the lab personnel who helped our students to navigate the last 10 weeks. Mentors and lab people, our PIs are instrumental. Without them, we would not be able to do this. And it is not easy. It requires time and commitment. Before I go and say something about the program itself, I would like to introduce Laura. She represents the leadership of CHLA. She is the Chief Operating Officer and the President of CHLA, and in her busy day, she took out some time. Thanks again for coming. Laura.
Thank you. Thank you so much. Hi. Good morning. It's such a pleasure to be here. I have been looking forward to joining you all week. Can we give a hand to Dr. B for all the amazing work that he does? Thank you for your longstanding commitment to nurturing future leaders in science and medicine. You are amazing, and what you do is really stellar. I'm so honored to be here today for the symposium to celebrate the incredible achievements of this year's group of young scholars. At CHLA, our mission revolves around creating hope and building healthier futures. And while many people know CHLA for the world-renowned pediatric care we provide to hundreds and thousands of children every year, another really essential part of our mission is about unlocking the potential of amazing youth throughout the communities that we serve. And programs like the Chuck Lorre Research Scholars Program are really a perfect embodiment of this. This program represents a really special convergence. It brings together education, research, and community, all elements that are embedded in CHLA's mission. And it's not just about academic excellence. It's about investing in the deep well of talent that we see in youth across Los Angeles. It's about nurturing the next generation of scientists and healthcare professionals. And it's about encouraging and empowering young scholars to dream big about changing the world. And we really need you to dream big right now. This is a really important time for that. So today, we are so excited to feature presentations from the 2025-2026 Chuck Lorre Research Scholars Program class. We have 14 talented college students who've spent their summer conducting biomedical research here at CHLA and our affiliated organizations. And I want to just take a moment to address our scholars directly. I got to meet some of you this morning. I'm a huge fan. The potential the energy, the optimism that you represent and that you bring to what you're doing is just, it's amazing. You first came to us as LA HIP interns in high school. Remember when you were really young back then? And now you're coming back as Chuck Lorre research scholars. And through all of it, you've brought dedication, curiosity, work ethic, and commitment to the time that you've spent with us. You are not only advancing your own futures, but you're setting the stage for breakthrough discoveries that are going to not only change the lives of people, they're going to change the world. And so what you're doing it's a long game, I know, but it's also going to make a big difference. So I want to also recognize and acknowledge, I got to meet some parents and family members before we got started. Nice work. Your support, really. Young people are so influenced and benefit so much from adults who support them, encourage them, challenge them to do better. I remember my parents always said to me, we want you to do better than we've done, right? We're the beginning of something and we want you to do better. And so I know none of this happens without family and friends and loved ones. So congratulations. And of course, none of this would be possible without the generosity and visionary support of Mr. Chuck Lorre and the Chuck Lorre Family Foundation, helmed by President and Chief Giving Officer Tricia Cardozo. Thank you to our colleagues, friends, leaders for being here with us today and for everything that you've done. This is an amazing expression of your vision, and it's such a pleasure to have you with us today. As we hear from our scholars today, I'm going to encourage everyone to reflect on the vital role that each of you has played in inspiring this next generation of scientists and healthcare leaders. I see many of our leaders here today. Thank you for being here. I know we've got mentors and lab buddies and all sorts of people on the line with us. So the LA HIP and the Chuck Lorre Research Scholars Program represent a unique tag team that is providing an unparalleled pathway of opportunity for brilliant young minds, and we are so glad to be part of the journey with you. I'm so excited to learn from you all today. I'm not going to understand a lot of what you're talking about, but I know I'm going to learn a little bit more. I'm going to be a little bit smarter after this is over. So, congratulations and thank you. And I think I'm inviting Hannah Song, our Chief Opportunity, Integrity, and Impact Officer.
Just a few short words. That's a long title now. Good morning, everyone. My name is Hannah Song. Throughout my career here at CHLA, Caltech, and many other higher ed institutions, I've been really lucky to witness firsthand how true opportunity given to the broadest population is an absolute necessary condition for innovation and scientific discovery. Studies have shown time and time again that innovation is only possible when there are different perspectives, experiences, and identities represented. Look around this room. I'd say we've achieved this necessary condition for scientific excellence, especially for solving complex problems in pediatric medicine and biomedical research. When we create authentic opportunities for voices from all communities to contribute to science, we don't just transform individual lives. We strengthen and transform the integrity of our research enterprise and amplify our collective impact in all the communities we come from and care about. I also want to take a minute to acknowledge with deep respect the extraordinary support systems that have brought you here today. Your families, teachers, lab mates, mentors, they've provided the encouragement, sacrifices, and unwavering belief that's made this opportunity possible. As a first-generation immigrant student myself, I know firsthand what it means to be the beneficiary of such sacrifice, love, and hope. It's an honor to represent your community here today in this space. I hope you take some time to thank and acknowledge those who've guided you and believed in your potential and success. As you've all experienced, the path to scientific discovery is rarely walked alone, nor is it always fun and games. Your presence here represents not just your individual achievements, but the collective hope and investment of entire teams, labs, and communities who understand that when one of their own succeeds in science, it opens up doors for many others to follow, creating a ripple effect of discovery that will impact generations to come. So thank you for your dedication, for your commitment to research, and without further ado, I just want to turn the podium over to the first presenter today. I want to reiterate how proud we all are to be in this space, learning from all of you. Thank you.
Thank you, Dr. Song. So now I want to shift gears a little bit, and I hope I don't get too much in trouble with the CHLA Foundation, but 20 years ago, I presented an idea to the leadership of the Institute. And a couple of weeks after that, Linda Antonioli, who was the liaison between the Institute and the CHLA Foundation, she stormed into my office and said, you know, you have to come with me downtown next week. We're going to have to meet some potential donors. But you can't come in jeans. And then she gave me a complete rundown of how I have to dress and da 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 da. And she didn't tell me I have to get a haircut, but she wanted to. Okay. So, anyways, we went downtown. On the 52nd or 53rd floor, I was introduced to two people. That was Ted Samuels and his wife Lori. And we were sitting in a small conference room. There was even some little sandwiches there, but I was too nervous to eat. And Ted, without really chit-chatting, he went straight into the discussion and asked, so Emil, tell me, what is it that your ideas are? Why do you want to do this? Who's going to participate? How are you going to do this? Where are you going to find the students? Who pays for it? Okay? And I was reasonably prepared. I may be a little bit naive at the time, but I was reasonably prepared. But then he asked me a question that I was not prepared for. He asked me, and I paraphrase it, so Emil, if you do this, how do you know you're successful? What are your metrics that you're going to go and say, this program is successful? And it blew me away. And I must admit, to this day, I'm not so sure whether I have a good answer for this. But Ted, at the time, understood much better than I did. And I'm very honest about it. He understood what this is all about. He is an investor. He knew that you have to invest in young people. And he knew that this is not an investment with a short-term return. This is long-term. And if I come to introduce our Carolina Herrera, it took 12 years to get her PhD. So Ted, over the time, was not only some sort of a funder for the program of LAHIP, but he became a coach to me. He became a mentor to me. He took me under his wing, and I remember distinctly our 5:30 a.m. phone calls that I had with him, because he was on the East Coast, and he had 9 o'clock meetings, so I had to be in time here. And we talked about the program. We talked about where the shoe hurts, and he was there for me. And Ted, I hope we can see you now. Can you bring up Ted Samuels, please?
Can you hear me?
Yeah, we would like to see you. Yeah, here he is.
Oh, no.
Thank you, Ted, for being here. I really, really appreciate it. I had a wonderful journey over the last 20 years. And maybe you can tell us a little bit about your aspect, your thoughts about the last 20 years.
Well, thank you, and thank you for the introduction. I'm not quite sure I know who you were talking about, but just to echo everything that's been said already, congratulations to the students and to the family. It is it's an incredibly exciting and humbling day for all of us. And we're looking at the potential and the impact that you have and will have on communities and on science. And it really is mind-blowing, for lack of a better word. And Emil, I promise you won't get in trouble with the foundation. When I talked to Dr. B about saying a few words today, I also thought about 20 years ago when he walked into our office and talked about his vision and his passion for getting students from underrepresented communities aware of and involved in science and innovation and medicine and medical research. And it was simple and profound, not easy to execute, but there's not a better, there wasn't then and there's not now, a better, more capable institution, I think, in the world than CHLA to create this and execute it and get people involved from the hospital and all of us rowing in the same direction. So in thinking about what to say today, like many of you, I went to ChatGPT and looked at the mission, and Laura already talked about it, but it's a very simple mission, create hope and build healthier futures, but it's really complex and complicated, and much more complicated in today's environment than it's ever been. But the pillars of the mission and how we fulfill the mission, there are four. Caring for children's, teens, young adults, families, and each other, advancing knowledge through cutting-edge pediatric research and medical breakthroughs, preparing future generations of pediatric healthcare providers for the extensive medical education and residency programs, and supporting communities, and especially the underserved communities. And these pillars are fully embedded in the research programs that Dr. B dreamed about, had a vision about, had a passion about, created and nurtured. Along the way, it's been a journey. The first LA HIP program just started with the summer, but then very quickly we realized that there was much more that we could and should and needed to do to help students get into science and research. And so the next iteration was creating a robust college prep course and getting ready to get through the college applications, which is a daunting task in itself. Ultimately, we created LA HIP, or Dr. B created LA HIP 2.0 to keep the students in college engaged in scientific research, being part of papers and documenting achievements, which is all very important getting to the next step, which is medical school and PhD programs. And since Dr. B walked into my office, he said, how do you gauge success? I think the simplest and most important way to gauge it is the number of people who have gone through the program. And this year, at the end of this year from LA HIP, we had 260 students go through the program. And in the first year of the program, Rich Cordova, who was the CEO of the program, came to talk to the students. And one of the students said, if I go to college, I'll do it. And Rich said, to his credit, it's not if, it's when. And I think one of the beauties of the program, one of the incredibly important parts of the program, is it enables, encourages students to aim high and realize and recognize their potential. And again, it's been so incredibly important, so incredibly powerful. And so while today is really to celebrate the accomplishments of the incredible Chuck Lorre research scholars, I think, and if it were a full screen, it's also to recognize and celebrate Dr. B's accomplishments. So I'll stand up to give him a standing ovation. I think we all should. And by the way, I know Emil that he hates this because he's incredibly humble, but it's important because it was his vision and his passion that started off. I also want to thank the mentors, and everybody is, I mean, it's a journey. The mentors, the advisors, the lab folks, everybody who's had a fingerprint in this program, and particularly for this program, the incredible generosity of the Chuck Lorre Foundation, Family Foundation. Having you partner with us, gives a permanence and a potential to this program. And finally, the first year of the program, at the beginning, I welcomed folks and said, one of the really important things is to stay in touch with each other, with the community, and there's a strength and power in numbers. We have sufficient numbers, but just staying involved in the community and helping each other creates a network that has significant power. I mean, this is a difficult time for science, and staying the course, as Laura suggested, is really important, but this will ultimately pass, and the potential and what you will achieve excites us all, and as I said, humbles us all. But again, thank you, Dr. B, for your vision, your passion, your commitment, your persistence. The outcomes are just mind-blowing. And so thank you and congratulations.
Thank you. Thank you, Ted. And next time you and I are in the same town, let's get together and have a cup of coffee, okay? All right. Thank you, Ted. Before we get to the student presentation, I want to quickly say something about the program itself. So 10 years after inception of the LAHIP program, I did an outcome analysis of what happened to the student. Where did they go, what did they become for, and stuff like that. And it was heartbreaking. We had 100 students at the time who went to LAHIP, and very few stayed in science. Most went into non-STEM, some sort of major. Almost nobody went into any kind of degree granting program other than some sort of master's program in education and psychology and those kind of public health. And I realized I need to do something about this. And I thought we should have a follow-up program for LAHIP alumni only who are now in college to get them back into research. And, we didn't have too much money at the time, but I got lucky. I could convince the Doris Duke Foundation, big foundation in New York, to buy into my concept and say, listen, yes, let's do this pilot. And 9 years after the pilot, I did an analysis last year and think of the students who participated in the LAHIP 2.0, which I called it, and which is now the Chuck Lorre Research Scholars Program. Everybody who participated in that program stayed in science, majored with a major in STEM, continued to do research while in college, published, and went to PhD programs and med school. So to me, thank you. This is why the Chuck Lorre program is such an incredible, important program. Because we need to keep the students engaged in doing science. And we are incredibly lucky with Dr. Fry's leadership in LAHIP. Give him a hand. He supplies trained students for the Chuck Lorre program. That continuum, we both, Mark and I call it now from high school to grad school. This is an umbrella kind of protein. That continuum is very important. And we will continue to do this because we have now the financial foundation to do this without constantly writing some grants that may get cut. And I want to make sure Family Foundation. So Manal and I decided we don't want to moderate the sessions. So I have some stellar students. Who is the first moderator? Come on down here. Samantha Lopez is an incoming junior at Yale University. She was in the Chuck Lorre program last year. And then the Chuck Lorre program also is structured the way that some students is in the summer program and they want to continue. They actually can do research during their academic year. And the Chuck Lorre program is going to support that. So they don't have to go to some sort of a fashion store and then help out doing things, but they actually can do research. And so Samantha is going to moderate the session on neuroscience. Thanks again. Okay, and this is the thing. And if everything works, you have to press somewhere. I don't know, maybe here. Okay. Okay. All right. So we have to bring in our speaker from Hopkins. Jason, are you here?
Yes, I'm here, Dr. V.
Okay, good. All right, you're all set. I'm not sure you have to press. I think we can do it.
Okay. Hello, everyone. Thank you for coming. I would like to introduce Jason Perez, an incoming junior from Johns Hopkins University studying the gut-brain axis. Ooh. There we go.
Thank you so much for that introduction, Samantha. I'm actually waiting right now for my screen to be shared. I don't know if someone could help me with that. I think the slides are wrong. Yeah, I think it's
Jason,
showing your guys' screen.
I could click the slides forward for you if you just tell me when to go next.
Okay, awesome. Yeah, that sounds great.
You're on the title slide right now.
Okay, thank you very much. All right, so my project focuses on comparing semaglutide-evoked activation in the nucleus of the solitary tract and area postrema in the brain of genetically distinct mouse strains. Could you go to the next slide, please? Thank you. So currently, 43% of adults are considered overweight worldwide. Adult obesity has more than doubled since 1990, and adolescent obesity has quadrupled. This is important because obesity is associated with things like heart disease, metabolic disease, and type 2 diabetes. There are a variety of different treatments used to address obesity in patients, and one of which is an active ingredient in Ozempic and Wegovy, semaglutide. Semaglutide, or SEMA, is a glucagon-like peptide-1 receptor agonist, which basically means it can act like GLP-1 in the body and signals to the brain like GLP-1 does. GLP-1 is a hormone that signals for satiation and fullness to the brain. So in theories, injecting SEMA to the body would signal to the brain that you're full even without eating. 1 in 8 adults in the US are taking SEMA, but 10 to 15% of those people are not achieving significant weight loss and are even gaining weight on the drug. And sorry, the slide that before that is the slide I'm on now. Thank you. So SEMA has an effect in certain brain regions that are involved with feeding behavior and the feeling of fullness. These areas are the nucleus of the solitary tract, or the NTS, and the area postrema, or the AP. The way this works is that once SEMA is injected into the body and absorbed into the bloodstream, it makes its way to the AP and activates neurons, and that then signals to the NTS that makes its way to the AP and activates neurons, and that then signals to the NTS that produces the behavior. Next slide, please. So although we understand generally how SEMA interacts with the brain, we don't know why 10 to 15% of people are responding differently to SEMA. We want to understand what is different in low responders, or people that are not responding well to the drug, and high responders, people that do respond well in the brain regions that are involved with feeding behavior, the NTS and AP. It's difficult to study this in humans, so we use a mouse model with different strains of mice in order to model individual differences impacting how SEMA interacts in the brain. Next slide, please. Right. So the question I addressed in my project is, does the NTS and AP neuronal activity differ post-SEMA injection across genetically distinct mice? We hypothesize that low responders to SEMA will have less activation in the NTS and AP compared to high responders. In this experiment, we used 3 different strains of mice, C57s, DBAs, and NZOs. In a previous 3-week treatment of semaglutide, C57 and DBA mice lost weight on the treatment and were determined high responders. NZO mice, on the other hand, were considered low responders because they did not lose weight on the SEMA treatment. Next slide, please. So to investigate this, we needed to inject the adult mice with the semaglutide, collect the brains and fix the tissue, and then cut the brain tissue for the NTS and AP brain regions. We then stained the tissue with neurotrace blue, which helped visualize the anatomy of the cut brain tissue, GLP-1 receptor, which we could then analyze to see differences in the amount of receptor across the strains, and CFOS, which is a protein marker, it's that red channel down there, for neuronal activity, which we could manually count to also compare differences in amount of activity across the strains of the SEMA to the SEMA injection. We then image these slides and analyze the images. Next slide, please. So the following graphs depict the number of CFOS-positive neurons in the regions of interest. To preface, our sample size is low, and these results will be beneficial for power analysis to determine how many more subjects we need per group to reach significant differences. Based on our limited samples, we cannot conclude anything for certain, and as of now, the data is not significant, or NS, as shown in these graphs, in the NTS and AP. Next slide, please. So as for the density of receptor signaling for fullness, or GLP-1 receptor, again, due to our limited sample size, our data is not currently significant in the NTS or AP. Next slide. So due to our limited sample size, we cannot conclusively interpret the data, but is not currently significant, which does not support our hypothesis. In the future, the lab will increase its sample size and will include untreated samples that will be injected with a vehicle. We will pursue this research in the future because by identifying the differences in how SEMA interacts in the body and the brain across genetically distinct strains will help direct us in how to address better treatments for obesity. And the last slide, please. Thank you for listening to my presentation, and a special thanks to the Comedic Hara Lab that has mentored me and welcomed me into their labs for so many summers, and to the LA HIP students and Levitt Lab members that supported me throughout the summer. Thank you.
Thank you, Jason. That was a wonderful presentation. And now for our next presentation, we have Emily Morales, also studying something in neuro.
Good morning, my name is Emily Morales. This summer I conducted research in the Cortez lab, and my project was about investigating the geroprotective effects of exercise in the brain cortex. Aging, something we all don't look forward to. Aging is a biological process that affects every tissue in the body, including the brain. Researchers have identified 12 hallmarks of aging, which represent the key molecular and cellular processes that drive age-related decline. However, recent studies have shown that interventions such as regular exercise shows a promise in targeting these aging hallmarks. When you exercise, it has effects on several systems within your body, and my focus was looking at these systems within the brain. The underlying molecular mechanisms, particularly influenced by age, sex, and amount of physical activity, remain poorly understood. This led us to a research question. What neuroprotective benefits does exercise provide to the brain, and how are these effects influenced by age, sex, and exercise intensity? Based on previous literature, we were able to hypothesize that engaging in consistent physical activity provides neuroprotective benefits that counteract the hallmarks of aging in the brain. To conduct our experiment, we split our mice into 8 groups: younger adult and older male mice that were sedentary and exercised, and younger adult and older female mice that were sedentary and exercised. These exercised mice were given free wheels which tracked their daily activity, while the sedentary mice were given locked wheels as a control. After 4 weeks, we perfused all 8 groups and collected the brain cortex for immunoblotting to detect and analyze certain proteins within our samples. During these 10 weeks, I probed for several markers which targeted different hallmarks of aging, including altered intracellular communication and deregulated nutrient sensing. In this presentation, I will be discussing the highlighted probes which targeted for mitochondrial dysfunction, disabled macroautophagy, and the loss of proteostasis, systems that are vital for cellular health but tend to decline with age. LAMP1 is a lysosomal marker involved in the autophagy lysosomal pathway, which helps degrade damaged proteins and organelles. The symptometry quantifies protein levels based on band intensity, which we then graphed against wheel count. Due to the limited sample size, only pilot graphs showing significant comparisons are displayed to show how exercise intensity can affect brain protein levels. In males, we saw that LAMP1 expression was increased due to exercise, particularly in the younger males, where we see a positive running correlation of an R-squared of 0.96, something that wasn't visible in the older males. The female groups, however, had an opposite trend. Despite having higher baseline levels compared to males, they showed a decrease in LAMP1 due to exercise. This data shows a sex-dependent molecular response to the exercise in the brain's lysosomal pathway. You've probably heard this hundreds of times, but the mitochondria is a powerhouse of the cell. If that's the case, then TOM20 is like the door to the powerhouse, as it helps import other proteins into the mitochondria and marks for mitochondrial abundance. In the young males, we see how exercise significantly increased the overall TOM20 levels, but we saw a decline in TOM20 with more exercise, with these mice having an R-squared of negative 0.8. In the old males, we also saw an increase of overall TOM20 levels, which strongly correlated to running distance, having an R-squared of 0.99. The female groups also showed an increase of overall TOM20 levels. However, they showed no significant running distance correlations. What this means is that TOM20 was increased in all groups due to exercise, but the relationship to the amount of physical activity varied based on sex and age, showing subtle differences in mitochondrial regulation. PINK1 is a mitochondrial quality control kinase that helps remove damaged mitochondria. At baseline, we see that all females presented with higher levels of PINK1, suggesting a more active mitochondrial quality control. The only groups that were upregulated due to exercise for PINK1 were the young mice. However, there were differences in sexes between the running distance correlations, with the young males having a negative R-squared of 0.8, while the females had a positive R-squared of 0.82. For the old mice, we saw a decrease in PINK1 due to exercise, with only the old males having a significant running correlation of an R-squared of 0.86. These results show a sex and age-dependent regulation of mitophagy, with females having more pronounced exercise-induced PINK1 responses. In conclusion, we were able to determine that engaging in consistent physical activity can target some of these 12 hallmarks of aging, but reveals sex and age-specific molecular mechanisms. In LAMP1, we saw this sex-specific regulation, with lysosomal activity only increasing in the male groups. And we saw differences in running distance correlations based on sex and age for TOM20, and only saw upregulation for PINK1 in the young mice. Our next steps for this project will be to confirm these trends and explore why there are differences in the molecular mechanisms between males and females. I would like to give a special thanks to the Cortez Lab, especially my PI and my mentor, Dr. Cortez. I'd also like to thank Dr. B and Dr. Manal for all the guidance that they've given me throughout the summer. And lastly, I'd like to thank the Chuck Gore Research Scholars Program at CHLA for this incredible experience. Thank you.
Thank you, Emily. And next, I'd like to introduce Leslie Cisneros, an incoming junior from UCLA studying peripheral nerve regeneration in mice.
Good morning, everyone. My name is Leslie Cisneros, and this summer I had the privilege of doing research in the Geschwin lab at UCLA under the mentorship of Juan Su. So my project focused on modulating GABA A signaling to promote functional repair in peripheral nerve injury. So a little bit of background, the peripheral nervous system is a communication network that relays information from the central nervous system, which is our brain and our spinal cord, to the body. And previous drug screening that has been conducted in the Geschwin lab has identified GABA A receptors as targets for enhancing axon regeneration. Therefore, my project aims to validate GABA A receptors agonists as therapeutic candidates for PNS injury. And this is very important because most of the time, PNS injury results in incomplete recovery, and it can be very painful. So for this reason, since we're focusing on the PNS, we focused on the sciatic nerve, which is the largest nerve in the human body. And as visual, the sciatic nerve runs from the bottom of our feet all the way to the bottom of our spinal cord. And we performed these experiments and this project on mice since we can't perform this on humans. So this led to our research question of, does treatment with GABA A receptor agonist increase neural outgrowth in mice compared to vehicle controls? And we hypothesized that treatment with previously screened drugs does indeed, which previously screened drugs targeting GABA A receptors does indeed promote neural outgrowth. So for my project, we focused on mouse models that were adults, and we first began by injuring the sciatic nerve, and we did this by using a scalpel and severing the sciatic nerve or using forceps to crush the sciatic nerve. Immediately after, we performed an intraperitoneal drug injection, and for every mouse that was injected with the growth-promoting drug, we also had a vehicle control. And as you can see, for this experiment, we had 7 vehicle controls and 7 mice that were injected with the growth-promoting drug. After 72 hours, we perfused and fixed the tissue with PFA, and then we dissected and cleared the whole sciatic nerve using a live canvas method, which allows us to image the entire sciatic nerve without having to cut it, which results in more objective analysis. After this, we tagged the sciatic nerve for things of our interest, and then we imaged and performed statistical analysis. So as you can see here, this is the sciatic nerve of our vehicle control mouse. And the white bar represents the injury site. So GD tomato represents the Schwann cells, which are important for myelination of the axon. DAPI represents the nuclei. The cyan represents neurons. And STMN2, which is the magenta, represents the newly grown neurites. So for my experiment, this is mainly what I'm focused on. So as you can see here, this is the injury site, and we measure the neural outgrowth up until here towards the distal end. So this is also a sciatic nerve of a drug-treated mouse, and it's the same exact thing. But one thing that you can notice is that we can't really see the injury site, and this could be for 2 main reasons. One, because there was a mistake in the embedding process or an issue with the confocal. So in conclusion, the tissue clearing method enables for more objective analysis of axon growth compared to traditional cryosection and immunohistochemistry methods. And the live canvas method shows more promising results for clearing the mouse sciatic nerves, allowing us for further measure growth promoting effects of GABA-A drugs. So some next steps would be to image the remaining sciatic nerves, which would be the vehicle and the GABA-A drug sciatic nerves, and perform statistical analysis to measure the neural outgrowth and compare the vehicle to the drug treated. And excitingly, we're also working with Malteni Biotech to use their ultra microscope blaze, which would reduce the imaging time by more than 50%, which is very exciting because this imaging process takes like 2 hours for just one sciatic nerve. And we will also be able to do 3D imaging. And lastly, I want to thank the Chuck Lorre Research Scholars Program for allowing me to be here today, as well as the Gershwin Lab and Juan Su, my mentor, and my 2 undergrads alongside me, and Grace Shin. Thank you.
Thank you, Leslie. That was amazing. Okay, so next is myself. My name is Samantha Lopez. I'm an incoming junior at Yale University, and I'm studying nerve regeneration in planaria. So over the course of this summer, I was studying the role of glia in planarian axon regeneration. Now, in the US alone, there are 18,000 new cases of spinal cord injuries annually, and as you can imagine, this is not great, especially because humans have a very limited regenerative capacity, and so individuals with these injuries often have motor and sensory disabilities that they just have to deal with for life, essentially. Now, this is not the case for planarians, which are a type of flatworm that are capable of regenerating any type of their body, or any part of their body, including the entirety of their central nervous system, as well as their visual system, which you can see labeled here to the left. They have the brain, this little arch-looking thing connecting the 2 eyes is called the optic chiasm, their eyes, these nerve cords. And we're interested in using them as a neural regenerative model in particular because we can use their visual system to test for functional recovery as well as just physical recovery of their nerves, their axons after injury. Now, there are 2 regeneration paradigms that I studied this summer. The first one being de novo regeneration, which just means from scratch, essentially, right? Say I cut off the head, how does it regrow the brain? Does it regrow the brain? And then we have axon regeneration, which would be more similar to repair or what you can imagine if somebody actually incurred a spinal cord injury. Now, glial cells have been shown to assist in axon regeneration in various other animal models, and so I want to know, how do glial cells impact axon regeneration in planaria? So that's our question. How does knockdown of glial genes impact nerve cord and optic chiasm repair, as well as de novo neural regeneration in planaria? And we hypothesize that knockdown of glial genes will hinder axon regeneration and repair. The genes in particular that we're interested in are Estrella, ETS1, IF1, and calamari. These are all glial cell markers or are just, co-expressed with genes that we think are associated with these glial cells in planaria. It's very specific and novel, and we're not entirely sure about everything yet, but that's what we think. And so for the methods, I started off by cloning and isolating our genes of interest, which we then used to synthesize double-stranded RNA, which we then fed to the worms in their little liver food that we feed them over the course of 3 weeks. And this essentially just turns off the genes, so that way they're not really functional in these worms. And after the 3 weeks, the genes should be pretty well turned off. And we go ahead and amputate the head, we fix the heads, let the worm regenerate. They're perfectly fine, it's fine. They regenerate for 7 days. They grew the head back, they regenerate. And then we fix those regenerated heads, and we use fluorescent in situ hybridization to visualize the remaining expression that's left over of the gene to compare, the gene expression. And we also use it to visualize the brain, and then we use antibody staining to visualize that axon bundle I pointed out, the optic chiasm, as well as the eyes. Now for results. First, we have the RNAi efficacy after the 7-day post-amputation. We have our wild type, which is our control for each of the genes. And that just means the regular worms, they didn't have any double-stranded RNA I fed to them. This is how it should normally be expressed. So Estrella, you would see this green here. That's what it would look like expressed in the brain and kind of going down the nerve cords. ETS1 expressed kind of around the periphery of the worm, very general expression. And IF1 and calamari also have a very similar expression, just kind of in the brain. Now, as you can see with our Estrella knockdown, there's much more reduced expression. Same thing with ETS1, IF1, and calamari. It's a bit harder to tell, but I hope to get clearer images in the future. Now for our heads, 7 days post-amputation, these are the regenerated ones. So they're impacted by the double-stranded RNA that I fed them. Our controls for these were just worms that were fed a different double-stranded RNA that should have no impact on genes that we're looking to study. We just use this to make sure that there's no difference that occurs due to the procedure itself, right? So this is what the brain should normally look like here in pink, and this is what the optic chiasm and the eye should look like. Okay, so Estrella, as you can see here, the brain looks very diffuse in comparison to the control and the other treatment groups. And for calamari, the optic chiasm appears very high up, close to the eyes. So those were two morphological differences that stood out to me kind of immediately. So 2 key takeaways from this. First of all, our glial RNAi did in fact work. The gene expression was reduced, and there appeared to be some difference that occurred. 2 morphological differences were observed in the brain of Estrella knockdown worms and in the optic chiasm of calamari knockdown worms. Going forward, I need to continue with some other experiments testing for repair of the nerve cords and optic chiasm, investigating potential upregulation of genes associated with axon repair, because what you just saw was de novo regeneration, right? And I need to finish imaging and then analyze the images to get actual quantitative numbers for this. And a special thanks to my mentor, Karina, and my PI from the von Wolfswinkel Lab at Yale University. You can see our baby worm there. And thank you to the Chuck Lorre Scholars Program for making this possible.
Thank you, speakers in neuroscience section. Thanks, Samantha, for handling it. Now I have the distinct honor to bring up Diana. Come on. Diana is a LAHIP alum from 2016. And I asked her to come when I was in Japan last fall. I think I got a phone call at 2 o'clock in the morning because she didn't know I'm in Japan and the time difference is so. And we talked about, she said she needs input and advice of her next step to get into the PhD program. And we had, I guess, a really frank conversation. This is Dr. B, okay? So, and she will talk about her journey today. Pay attention. She has some really interesting things to say. Thanks.
Hi, everyone. Yeah, my name is Diana, Before I start, I do want to congratulate all the scholars who are accomplishing all their research. It sounds pretty cool. Dr. B did say no tears, so bear with me. I was part of the LA HIP class in 2016. I graduated from John H. Francis Polytechnic High School in Sun Valley. And I will say that LA HIP was definitely my first real stepping stone towards a science career. Really narrowed my interest to regenerative medicine and bioengineering. And I did go to Occidental College because of LA HIP. Dr. B took us to a field trip to Occidental, and I fell in love. But when it came down to actually going to Oxy, I completely panicked and regretted my decision. I remember coming to Dr. B. I came to the Saban ready to defer my acceptance, and he just stopped me in all my madness and gave me 3 options, saying, you can defer, take a gap year, go to community college, or just suck it up and go to Oxy. You already made the choice. And at that time, I was distraught. I thought it was the end of the world. I didn't know what to do. But basically, I stayed at Oxy and don't regret anything because, I graduated with honors, joined the rugby team, was a part of a neuroscience research lab that I completely loved. And it definitely has shaped who I've become today. Unfortunately, the pandemic did hit, and I wasn't as motivated to continue school like that. It wasn't how I envisioned my academic career. So graduating Oxy, I didn't have a plan. I worked for my family's business. We have a Mexican restaurant, and I was just dealing with financials, helping with events, covering shifts. I was honestly exhausted all the time, and I told myself I'd dedicate time to job hunt or just find the next step, right? But that did take some time, and once I did lock in and complete my Fulbright application, I heard back from Kite Pharma, where I was a cell therapy specialist, and I was part of the manufacturing department, which left actually zero room for errors or mistake. I had to follow the protocol by the book. And I was missing that curiosity, the mistakes that are so involved in research. And so when I did hear back from my Fulbright grant, I was more than ready to go. I left to Spain. Yeah, thank you. I left to Spain, left my family for the first time because, I was just LA, San Fernando Valley. And just as I was familiarizing myself with my research, my PI, because we had a lot of conflicts, the culture, my roommates, my travel plans, I heard news that my brother passed away. So I had to come back, and sorry. It was hard, my motivation was not, I didn't have it. My heart was not in research. I came home, didn't know if or when I was gonna go back to Spain and finish the grant. I was just with my family. I wanted to make time for them and grieve and mourn, which I did. But I also knew that I didn't want to give up. So I did go back to Spain, and I finished my grant, presented my poster, did everything, but really my heart was not in it. And when I finished the grant, I came back home. I decided to take my brother's role in the family business, which was financial advisor, right? Or just dealing with the financials. But that responsibility turned into dealing with the catering events. And that one turned into covering people's shifts. And I was just everywhere and realized that I was just stuck in this same cycle. And until I put myself first, I wouldn't get out. So slowly but surely, I started to reach out. I started looking for programs and applying. I was exhausted, yes, but I decided to pick me for once. And the crazy thing is that the only school that I applied to out of California was the only one that accepted me. And I applied just like on a whim, right? But right now, I'm actually more than excited, very nervous to go to Oregon. I got accepted to the University of Oregon in the bioengineering department. Thank you. But yeah, I think this is the first time I'll be able to just kind of focus on my education, because I know that if I would have stayed in LA, it would have been so easy to just, let myself go back into the business, which is currently expanding, but I'm excited. And with all that being said, I just want to thank everyone for listening. And I do want to let people know that you decide if a challenge or an obstacle is going to make or break you. And if you really want something and you work for it, you just need time. They say that time, and it doesn't feel like that in the moment, but it really just is. But yeah, thank you.
Thank you for coming.
Thank you. Thanks, Dr. B.
Okay, Emily. So the next section is on some sort of computational biology and biomedical engineering. We have two boys here who will talk, but Emily will moderate the session. Thanks again, and please take the cursor when you talk up here on the microphone. Don't eat the microphone.
Thank you, Dr. B. The first person I would like to call up is Edgar Gomez, an incoming sophomore at Columbia University who studied lipid nanoparticles and gene delivery. Come on up, Edgar.
That was not the right button. There we go. Okay. Hello, everyone. Thank you all for coming. Today, I'd like to talk about one of the projects I worked on at the Chung Lab in USC, which was engineering lipid nanoparticles for attenuating polycystic kidney disease. So for those who don't know, autosomal dominant polycystic kidney disease, or ADPKD, is the most prevalent genetic kidney disease in the world. It impacts more than 12 million people worldwide, according to recent NIH reports, and arises from mutations in the PKD1 or PKD2 genes, where loss of the gene products of PKD1 or PKD2 will result in aberrant proliferation of collecting duct or CD cells, which will drive rampant cyst formation, these really nasty looking cysts, inflammation, and if left untreated, kidney failure. Unfortunately, current treatment options for ADPKD only offer minimal symptomatic relief and do not address the root genetic cause of the disease. So recognizing the current limitations in ADPKD therapies, our lab has been investigating the use of gene replacement therapy via lipid nanoparticles. Lipid nanoparticles, or LNPs for short, are self-assembled lipid nanostructures that importantly will encapsulate and deliver various therapeutics like mRNA, which could substitute the missing genetic material in ADPKD with greater efficiency and stability than free injections. Unfortunately, administered LNPs do not have very significant renal accumulation, meaning that administered LNPs will not usually deliver the required therapeutics to the diseased kidneys. So to combat this, we've looked at conjugating a targeting peptide to the surface of our LNPs to improve site-specific delivery and renal uptake. Targeting peptides are short amino acid chains that will preferentially bind to specific cell types, tissues, or cellular components. Essentially, they act as mini homing devices for our LNPs. So the specific cells that we're looking at targeting here are those proliferative CD cells or collecting duct cells. So knowing this, we want to find out whether functionalizing these LNPs with a CD targeting peptide would improve the uptake of encapsulated therapeutic mRNA to the kidneys compared to control non-target LNPs. We hypothesize that they will. So to answer this question, we first created Cy7 labeled LNPs encapsulating a marker M-cherry mRNA and intravenously administered them to ADPKD mice. 24 hours post-treatment, these mice were euthanized and their major organs were harvested for analysis through various means that would allow us to both assess the biodistribution of these LNPs while also verifying and validating the successful delivery of the marker M-cherry mRNA. First, by measuring the levels of fluorescent Cy7 that were attached to our LNPs, we're able to use the IVIS or in vivo imaging system to measure the total distribution of the LNPs throughout the different organs. Ex vivo imaging of the organs 24 hours post-treatment revealed that the CD-LNPs accumulated to a much greater extent in the kidneys than their non-targeting counterparts. In fact, they're more than 70% more effective at transfecting renal cells than their non-targeting control. Following imaging, these kidneys were processed into single-cell suspension, where the different cell types of the kidneys were stained, most importantly being the CD cells. Then these cells were sorted using flow cytometry, which was made possible by the different fluorescent molecules present in these samples. These results showed us that by looking at the levels of Cy7 present in these renal cells, we were able to confirm that the CD-LNPs were almost twice as effective at transfecting renal cells than non-targeting LNPs. Furthermore, the same samples highlighted a massive increase in the uptake of LNPs in the collecting duct cells, showing that the CD targeting peptides were working as intended. Similar positive results were also obtained when analyzing the proportion of the marker M-cherry mRNA that was actually transfected by the cells. These results show that decorating these LNPs with the targeting peptides drastically improved CD cell targeting and mRNA transfection, showing us that not only were these LNPs locating more disease cells in the kidneys, but they were also successfully delivering the therapeutic mRNA at a greater rate and at the same dosage. From the data collected, it's clear that conjugating a CD targeting peptide to the surface of these LNPs enhanced overall renal accumulation, direct them preferentially to the cis lining CD cells, and overall improve the therapeutic efficacy of mRNA treatment via lipid nanoparticles. And we've already seen this to be true in previous data conducted in our lab, showing that CD-LNPs encapsulating PKD2 mRNA, which is one of the aberrant mutations in the ADPKD, actually reversed cyst growth and restored a lot of renal function to these mice. In the future, we're hoping to continue to test the efficacy of CD-LNPs with this therapeutic PKD2 mRNA in more genetically diverse mouse models. I'd like to give a special thanks to the Chung Lab, especially my mentor, Josh, for being so incredible. I'd also like to thank the Chuck Lorre Foundation for providing the funding for the summer. And lastly, to the LA Metro and my parents for getting me to and from work in a timely manner. Thank you.
Thank you so much, Edgar. Next up, we have Samuel Sosa, an incoming sophomore at UPenn, also studying computer modeling of pattern formation. Come on up, Samuel.
Perfect. Good evening, or I guess the morning, everyone. Oh, actually, let me switch this. Perfect. Yeah, as Emily said, I'm Samuel Sosa. I'm a rising sophomore at the University of Pennsylvania. And this summer, I spent my research at Caltech under the Winfrey Lab with the project titled Neural Cellular Automata for Sequential Pattern Formation. So I want to pose a question. So first, if we think about a single cell, we could also say that, or think about how it fully grows into a human, much like us. So in this example of an embryo, we start at a relatively simple state here towards the left with little to no cells. And then as we progress here, as we go along, we can see that the cells multiply and they grow and they advance to a more complex stage of the embryo. And what this shows is that the cells are able to generate or self-organize themselves in a way that it doesn't require a global coordinator or a global blueprint, and it just functions at a local level and it grows to something big. And the bottom line here, as I mentioned before, that the cellular self-organization drives tissue and organ formation. So this inspired my question. Can local cellular interactions guide the formation of bigger, more complex global patterns or structures, better said? And can this generate multiple patterns and to emerge sequentially? So my hypothesis was that global modeling local interactions can drive complex self-organization, and at the same time, sequential cues can enable multiple patterns. So my focus was more on the computational side. And here on the, I want to start with the right. Here in this butterfly life cycle analogy, we can see that we start from a egg, or we could say as a seed, and then we go from the larva to the pupa and to the butterfly. And each stage we have distinct pattern. And in this example, I want to highlight that in nature, we're not just given a copy of, say, a butterfly, and we say, okay, copy this. There are cells that have to learn and to self-organize themselves in order to develop into a more advanced stage. So in the model training loop here, I ran this for a couple dozen steps, to 4,000 steps. So in our computational model, we start off with a seed, and then we let it grow to our first pattern. And in our first pattern, it's supposed to resemble some sort of lizard, just for reference. And then in the second pattern, we're supposed to resemble some sort of butterfly. And as you can see here, as we continue in steps, from step 40, we can see that the pattern is not very apparent, but then as the model learns to self-organize the cells, which are represented by pixels, we can see significant progression from step 400 to step 4,000, in which we can see visual success. So the results here, towards the left, you can see that it was visually successful, but we quantified this and we saw that our training loss, or in other words, our error rate, if you will, it's converged through the thousands and thousands of steps. And what this just means is that our model is able to adapt to new information. So again, what this means is that if you give a new image, it's able to produce a new image, and so on. And this is really important because we know we can model back-to-back patterns. So before we could just model one pattern, but now my research pushed it further, and okay, we can now model more than one pattern, in this case, 2 patterns. What about 3 patterns, right? So here we have 3 patterns in which we start from a single seed, and then we develop to a gecko lizard-like animal figure, and then a butterfly, and then a pretzel. So again, this may be a little confusing, but the reason I used these types of emojis is because in the original paper, that's what they used, and this is really representative of how we could use images and patterns to provide this sequential pattern training. And then, but again, what's the purpose of all this? So this is important because we can model real-life sequential patterns, like we saw the butterfly analogy and our embryonic development example. So the key takeaway here is straightforward. We can produce computational cells and produce this model that can self-organize themselves into more complex structures like these patterns using only local interactions without a global coordinator building everything. And we can use sequential cues or activators if you want to compare it to a more concrete biological analogy that can provide multiple patterns for more time. And this just provides a simple but very instructive model of cellular development. And for future work, to pass this into real life, but for biomedical researchers, this means that we can give the competition model a 3D target of a tissue or an organ or whatnot, and then let it replicate the structure through a series of self-organizing cells. Again, lastly, I want to give a special thanks to the Winfrey Lab for having me at Caltech this summer, and for the Chuck Lord for providing the funding and for making this summer a really amazing summer. Thank you.
That's the future. Thank you, Sam. So I want to introduce our next moderator, Juliana. Juliana is an incoming junior at UCLA. She was in the Chuck Lorre program last year summer, and she continued in the lab during the academic year. So She's going to moderate, and she's going to be the last speaker on the session. Thanks again. Okay.
Hi, everyone. I'm going to introduce the section on stem cell research in regenerative medicine. So first up, we have Anna Diop. She is an incoming sophomore at UC Irvine, and her project will be on limb regeneration in Drosophila.
Good morning, everyone. Thank you for having me. So to provide some context, my lab works in the field of systems biology, which seeks to understand the bigger picture of biological systems by putting the pieces together, studying how different components interact to produce the behavior of the whole. Currently, our work focuses on the model organism Drosophila melanogaster. Untreated flies do not normally regenerate. Instead, the stump forms a scab, seen in this dark mark here. When flies were treated with leucine, insulin, and glutamine, many flies did not form a scab. Instead, they began to regrow. As you can see in the images here, at 0, 14, and 22 days post-amputation, there is a clear regrowth of the tibia. This phenotype was first observed using bright field microscopy, an imaging method, and ongoing work is now characterizing the regeneration process at the cellular level. Based on the literature, we know that this specific strain of Lactobacillus brevis can produce two key metabolites, acetate and ornithine, through fermentation. Acetate is produced when the bacteria ferments sugars like sucrose and while ornithine comes from fermenting amino acids like arginine. My research question was, if we feed Lactobacillus brevis to adult flies, does this change the levels of acetate and ornithine in the fly? Leading us to the hypothesis that L. brevis will increase both metabolites in the flies. The idea is that if ornithine and acetate concentrations increase in the fly, they could act as signals that influence tissue regeneration, similar to how leucine, insulin, and glutamine did in the 2021 study. For this project, I used two main assays, an acetate assay and an ornithine assay. First, the feeding protocol. I inoculated MRS media with Lactobacillus brevis. After a 2-day incubation, I isolated the bacteria and prepared two groups of Cantonese flies, one fed with L. brevis and one untreated control group. 1 day after feeding, I homogenized the flies and collected samples for each assay. For the acetate assay, using a commercial kit, I prepared a separate set of standards, added a colorimetric master mix to the samples, and again measured absorbance at 550 nanometers. And for the ornithine assay, we prepared standards and reacted both the standards and the fly samples with 9-hydrin, then measured absorbance at 515 nanometers to quantify ornithine levels. Before I could measure acetate and ornithine levels in flies, I first made standard curves for each assay using absorbance values from known concentrations of acetate or ornithine standards. Both curves had R-squared values greater than 0.9, indicating my measurements were consistent and the equations were reliable for calculating unknown concentrations. Once I confirmed the assays worked, I used these curves to calculate metabolite levels in the flies. Each bar shows the average result from 6 samples. Each sample came from 12 flies that we processed together into one homogenate. We repeated this process in 2 separate experiments to make sure the results were reproducible. We used a tease test, which is a statistical method that compares the averages of 2 groups to see if the difference between them is likely due to chance or to the treatment. As shown for the acetate assay, there was no significant difference between treated and untreated flies. The percent change in acetic acid production between the untreated and LB fed groups was 0.61%. For the ornithine assay, we saw a significant increase in ornithine concentration in the LB treated flies compared to controls, leading to a percent change in ornithine production of 56.1%. So acetate levels did not change under our experimental conditions. Ornithine levels did increase in correlation with LB feeding. From our results, we see that there was no significant difference in the acetate assay. However, the ornithine assay showed a substantial difference, which points towards a possible role for L. brevis in influencing ornithine-related metabolic pathways. The long-term feeding studies will help us understand how L. brevis affects ornithine metabolism over time, capturing delayed or cumulative changes. This foundational knowledge is crucial before moving to the long-term goal of amputating flies to test whether L. brevis feeding influences limb regrowth. By establishing nutrient changes first, we can better interpret any effects on regeneration, determining whether they are likely linked to ornithine metabolism or if other factors might be contributing. And a special thanks to everyone, the Chuck Lorre Research Scholars Program for supporting my work, the Guntoro Lab at Caltech, my PI, Dr. Leah Guntoro, Judah Bates, and as well as Dr. B and Dr. Manal Taba for helping me throughout the summer. Thank you.
Okay, I want to say very special thanks to Anna for her amazing presentation. Next up in this section, we have Donna Nava, who's also an incoming sophomore. She goes to USC, and her research will be on craniofacial development in zebrafish.
Good morning, everyone. My name is Donna Nava, and I am a rising second-year student at the University of Southern California. Today, I will be discussing the role of the PIDX gene family in neural crest mesenchyme for zebrafish skeletal development. So the PIDX gene family has been linked to several developmental disorders. For example, PIDX1 has been associated to micronathia, which refers to the abnormal formation of the lower jaw being smaller than normal. On the other hand, PIDX2 and PIDX3 are both involved in eye development. PIDX2 has been primarily associated with Axenfeld-Rieger syndrome, which can lead to glaucoma, while PIDX3 is associated to congenital cataracts or cloudiness in the lens, as well as anterior segment dysgenesis, which affects the front part of the eye. Interestingly, previous research in our lab has shown that PIDX genes are active in the frontal nasal region, which is the area from the bottom of the nose to the top of the head. This raises an important question. What cell population is responsible for shaping these craniofacial and ocular structures in the first place? That's where cranial neural crest cells come in. Cranial neural crest cells are a cell type crucial for embryo development, differentiating into numerous structures, including mesenchyme that give rise to bone and cartilage. These cells are characterized by their migration patterns, starting from the posterior of the embryo to the particular location of the structure. The involvement of cranial neural crest cells in craniofacial development, combined with PIDX gene structures, expression in these cells suggests that changes in PIDX function could play a pivotal role in shaping the frontal nasal region, which leads us to our research question. Do PIDX genes affect frontal nasal development? And our hypothesis that states the loss of individual PIDX genes will lead to the malformation of the frontal nasal domain. In our lab, we study these processes using zebrafish, whose translucent chorions and embryos make it possible to track development in real time and visualize any defects. Now, in order to determine how each PIDX gene affects the development of the frontal nasal domain, my mentor started with using CRISPR-Cas9 to knock out or delete the gene, establishing zebrafish lines with transcriptionless alleles for PIDX1, 2, and 3 individually. We started with having heterozygous fish, and through in-crosses would receive a predicted amount of 25% wild-type fish, 25% mutant fish, and 50% heterozygous fish. Next, we would perform a skeletal staining procedure where Alcian blue stained for cartilage and alizarin red stained for bone. This procedure was done on fish 6 days post-fertilization, since it's the time when bone starts to form, allowing us to see these structures without the tissue being too dense for staining. Imaging and phenotyping were then completed, which would limit bias, since by this point genotype remains unknown. Once imaging is completed, genotyping is done through PCR and gel electrophoresis. Before we begin to discuss our results, it is important to note that Meckel's cartilage is a temporary cartilage structure that supports early jaw development, while the jaw refers to the permanent bony mandible. For our wild-type Alcian alizarin stains, which is no mutations at all, We see a fully fused Meckel's cartilage, which is this area here, a straight early jaw, as seen here, and both lens remain intact. Now, for all three of our PIDX mutants, show a lack of Meckel's fusion, as seen through this gap here. And curvature in the developing jaw. Compared to our wild type, this jaw is straight and all of these show significant curvature, with this being more prominent in PIDX3. As a reminder, the jaw malformation is observed in patients with PIDX1 mutations such as micronathia. In PIDX2 and 3 mutants, we see that in PIDX2, our lens appear to be faint or reduced in size compared to our wild type, while the PIDX3 mutant shows a very faint lens. For both PIDX3, 2, and 3, Meckel's cartilage develops at a lower position, as seen through this gap here, compared to our wild type, with this displacement being more pronounced for PIDX3. To conclude, we confirm that PIDX genes are expressed in neural crest cells and visualize a clear pattern across the 3 homozygous mutants, which is the malformation of the jaw. Future work may include determining the mechanisms behind how PIDX genes affect jaw formation, which, as a reminder, doesn't form part of the frontonasal domain, or determining if there is a reciprocal relationship between the PIDX genes, where there is a possibility of compensation when one gene is knocked out, causing the upregulation of a PIDX gene. Lastly, we can explore other genes in the frontonasal domain area. I want to take the time to thank God, to thank the Crump Lab for having me this summer, and the Chuck Lorre Research Scholars Program for this amazing opportunity.
Okay, thank you again for that amazing presentation. Next up, we have Kaylee Oxner. She's also an incoming second year at Brown, and she will be explaining her project about craniofacial development in mice.
Hello everyone, my name is Kaylee Oxheimer, and today I will be talking about my project on investigating Alagille syndrome by characterizing the impact of Notch2 on tendon development in mouse jaws. That's a mouthful, I know. But before I begin, I want you to all imagine that you're looking into a mirror. What do you see? Probably your face, right? I'd be surprised if you looked at your elbow. Our faces are central to how we perceive ourselves and how others perceive us. But certain conditions affect these perceptions by altering the features we see in the mirror every day. And Alagille syndrome is one such condition. To be more specific, Alagille syndrome is a rare developmental disorder that impacts multiple organ systems like the liver, producing the jaundice we see in this patient here, as well as the cardiovascular system and the face, which features a triangular-shaped jaw, deep-set eyes, and an underdeveloped, flattened midface called hypoplasia. What we want to know is how this phenotype develops in patients. Previous studies have shown that patients with Alagille syndrome often have mutations in the Notch2 gene. But what is the Notch2 gene? Well, Notch2 is a gene that codes for a protein in the Notch signaling pathway. A signaling pathway is a way for cells to communicate through 2 main parts. A ligand, which you can see in this image here, sticks out of one cell, and a receptor, which sticks out of another cell. Once the ligand binds to a receptor, a signal is transmitted throughout the cell with the receptor, producing a change inside the cell. In the case of the Notch signaling pathway, there are 4 receptors, and the Notch2 gene codes for proteins forming the second one. Once the ligand binds to this receptor, a protein complex is released called NICD. One protein complex is released called NICD that binds to genes that regulate cell fate. Essentially, this means that the Notch signaling pathway is responsible for regulating development of specialized cells during development, causing the specialized tissues like connective tissue and muscle tissue. As you can see in this image of where Notch2 is expressed in a mouse jaw, there's elevated expression of Notch2 in specific regions like the condyle, C, and the angular, A. There's also elevated expression in the tendon, T. And this region is particularly interesting because if mutations in the Notch2 gene are associated with Alagille syndrome, then since there's elevated expression of Notch2 in the tendon,
Maybe it is deformities in the tendon that lead to the phenotype we see in Alagille syndrome. So our question is as follows. Is the tendon and jaws where Notch2 has been deleted different compared to normal controls? We hypothesize that loss of Notch2 function disrupts tendon development in the jaw. To investigate our question, we use the Wnt1-Cree-LOXP system in mice. The Wnt1-Cree-LOXP system is essentially a pair of molecular scissors, allowing us to cut out genes in specific cells. In our case, we want to cut out Notch2 in cranial neural crest cells, which contribute to the formation of tendons in the face. We perform this technique in mice because the genetic system we use in mice is a good model for human disease. Once we breed offspring through the system, we collect them at 2 days old. Then, we genotype to find mice with deleted Notch2, our mutants, and mice with a complete wild-type genome, our controls. Then, we preserve and embed mice skulls in paraffin wax in order to section them using the microtome. We then stain these sections to visualize specific skull tissues and analyze our mutants in comparison to our controls. After completing this process, we produced these images. We used HPQ staining, so in this picture, the blue is cartilage and the pink is muscle. While there are a few differences among the samples, we can see that the images are overall very similar. For example, in the shape of the condyle, you might notice that these two are pretty different, but that's likely due to the fact that there's differences in sectioning plane, and sample morphology, and sample preparation. Zoning in more closely on the tendon, there does not seem to be a major difference between the two. When analyzing the tendon in terms of attachment to the angular, length, width, and shape, there do not appear to be any differences. While there are subtle differences, such as on the right-hand side of this image here, there's again not sufficient evidence to suggest that Notch2 is responsible for tendon development. However, while we did not see a difference between tendons in the jaws of mutant mice and control mice, a benefit of our experiment is that we can analyze different parts of the skull at once. In comparing the top of the skull, called the calvaria, in mutant mice to control mice, we found some interesting results. These images are of the coronal suture, which is demonstrated here. The coronal suture is a ligament that connects two skull bones. So, as you can see in the circled region of the control mouse suture, there is a gap between the yellow parts, which represent the bones. This is why we see a gap in the image here. However, in the mutant mouse suture, there is no gap, as there's yellow on both sides here. This means that Notch2 has a clear impact on sutures, which may suggest that it plays a unique role in ligament development rather than tendon development. So, to conclude, Notch2 does not have a pronounced impact on tendons in the jaw based on analyses of shape, length, attachment, and width. This may be because a different receptor, specifically Notch1, may replace Notch2, since both are active in the jaw domain. Since this allows Notch signaling pathway to function normally, this may cause normal development of the tendon. This also may be because Notch2 impacts other parts of the skull, which, as I've discussed previously, includes the sutures. So our next steps are to analyze the role of Notch2 on such sutures and on ligament development. Thank you to the Merrill Lab and to my mentor and PI, and to the Chuck Lorre Research Scholars Program for this experience. Thank you.
Okay, again, thank you so much to Kaylee for your presentation. I will be the next presenter. My name is Juliana, and I'm an incoming third year at UCLA. This past summer and over the past year, I've been working in the lab of Dr. Lee on synthesizing skull regenerative biomaterials. Okay, first, a little bit of background. Why is it important that we're studying this? Skull reconstruction is required in 13.8 million cases annually. The most effective method is an autologous bone transplant, seen here, which uses the donor's own bone from a different region of the body. But this is pretty limited by donor site morbidity and defect size. The second best option is an alloplastic implant. These include titanium and different plastics, but they often lead to higher complication and failure rates. In order to combat this problem, my lab has been working on developing an MC-GAG scaffold. This scaffold is composed of type 1 collagen, chondroitin 6 sulfate, and nanoparticulate calcium phosphate, and it actually promotes the development of bone itself, rather than relying solely on the implant. Okay, what specifically am I studying? The first part of my hypothesis includes tagging the MC-GAG scaffold with a PEG-RGD peptide. This is a protein motif that binds to cell surface receptors and integrins in order to increase cell adhesion. I hypothesize that functionalizing the MC-GAG scaffold with PEG-RGD will enhance the migration and uptake of human mesenchymal stem cells. We're specifically using HMSCs because these are cells that have yet to differentiate. This means that we can encourage differentiation of these stem cells into bone or osteoblasts. The second part of my hypothesis includes tagging these stem cells with a poly-L-lysine conjugated nanogold. Because the material is pretty opaque, it can be hard to visualize the cells through the scaffold. Because of this, I hypothesize that tagging these cells with nanogold will enable us to instead visualize the nanogold itself through different 3D methods such as confocal imaging. Okay, now, how am I doing this? After preparing the two different scaffolds, which are the MC-GAG and PEG-RGD, we conjugate the nanogold with poly-L-lysine in order to change the zeta potential. We seed the different experimental groups, and we measure efficacy using a WST8 assay. This uses absorbance values to measure cell viability and proliferation, or essentially how much the cells are able to grow. I also use an alizarin red staining, which, as someone mentioned earlier, measures bone or calcium deposits. Next, we did a transwell migration assay to measure migration of the cells through the different scaffolds, and I imaged this using confocal imaging. Lastly, in order to characterize the nanogold after conjugation with poly-L-lysine, I used a dynamic light scattering technique. Okay, now we can get into the results. The nanogold, when we receive it from the manufacturer, is conjugated with a citrate for stability, but this has a negative zeta potential or charge. In order to increase uptake of the nanogold by the cells, we must conjugate it to have a positive potential. And to do this, we used poly-L-lysine. Using the dynamic light scattering, we were able to measure the shift in zeta potential from negative 56 to positive 6.4. As you can see on the next slide, the uptake of the nanogold is visualized by the black you see in the cells. Since there's a lot of black, we were pretty successful with the conjugation, and this means our cells were effectively able to absorb the nanogold. Lastly, on this slide, I have the results of our alizarin red staining. Media is how we grow our cells. It contains nutrients that our cells need. Basal media is just nutrients alone, while osteogenic growth media contains growth factors that promote differentiation of the cells into bone. In both the control and the nanogold, the amounts of red are pretty similar. This means we can use it as a tagging mechanism, since it doesn't really change the results of our experiment. Next up, I'm going to go into the scaffold, and specifically the PEG-RGD MC scaffold. The WST8 assay uses absorbance values to measure cell proliferation and viability, which tells us how many cells we're able to grow. The red bar here is the PEG-RGD scaffold, which has the highest absorbance value, meaning it was successfully able to encourage proliferation of cells. Next here, I have the transwell migration assay, where we count how many cells are able to pass through a membrane and into the scaffold, which measures how well the scaffolds themselves are able to attract the cells. In order to measure this, I counted the nuclei on each of the membrane. And again, the PEG-RGD scaffold had the highest nuclei count, meaning it was best able to attract the cells. Lastly, on this slide, I have a confocal image. The green that you see here is an actin stain, which is a type of measurement we use to see how many cells are present through the scaffold. This uses a 3D technique, and since you can see a lot of green, this means our cells were able to grow pretty abundantly. While we're still in the process of analysis, the bottom shows a different plane, and it tells us the volume of the cells. Okay, what can we conclude from all of this data? The first conclusion I was able to make was that the PEG-RGD functionalization of the MC-GAG scaffold effectively enhanced proliferation and migration of the cells through the scaffold. Lastly, I found that the nanogold tagging of the stem cells was able to increase visualization using 3D imaging techniques such as the confocal. Next steps will be to continue analysis of the confocal imaging before we can continue to in vivo studies using rabbit models. Hopefully, after all of this, we'll be one step closer to actual clinical applications that will help real people. Okay, thank you so much to everyone who helped me this summer and to all of you for listening, as well as Dr. B for helping to run this program and Dr. Tabah for also helping.
It sounds like a first-year graduate student. Congratulations. So now I have the distinct honor to introduce Dr. Carolina Herrera. Carolina was in LAHIP 2012. That's a long time ago. She actually she worked in Dr. Polk's lab. I saw her presentation. She has some really cool pictures of her at the time in LA. Thank you for coming.
All right, thank you so much, Dr. B. It's such an honor to be with you all here today. 13 years ago, I was standing in this very room delivering my summer research. So I was very impressed to hear all of your research today. And so today I'll give you a little talk about my career journey. And so the title of my talk is From Curiosity to Discovery, How One Internship Launched My Career in Biotech. So as I mentioned, 13 years ago, I was standing here in this very room. And so I was feeling both overwhelmed and excited about the future. I had no idea that curiosity in science could lead me to a PhD and to my current role as a preclinical scientist at biotech. Here are a few pictures that I found from 2012. The very first time that I stepped foot in a lab was in Dr. Polk's gastroenterology research lab. And so I was amazed to see all of the machines and lab equipment that I've never seen in my life. And I wanted to try everything, and I wanted to run to do my first experiment. And so the first picture on the left is Dr. B actually taking a picture of me during my first Western blot. The pointer, yeah. So right here, I was a little caught off guard, but I was very happy to do science here. And ever since LAHIP, during LAHIP, I learned how to think critically and how to be more of an independent scientist. And this is all thanks to Dr. B and also Dr. Polk for guiding me and to helping me how to think critically and actually how to do rigorous science as well. And so these are things that have stuck with me throughout my entire career journey. After LAHIP, with the help of Dr. B and LAHIP 2.0, I went to UC Irvine, and so I'll walk you through in the next slides my career journey. But basically, I went to UCI, I did a lot of research there, and then I had the opportunity to do research abroad in Spain, much like Diana, and I also did a PhD at UNC Chapel Hill. That led me to a postdoc at Vertex Pharmaceuticals, and then that led me to my current role as a preclinical scientist at Capstan Therapeutics. So LAHIP really opened my eyes to a research world I never knew existed, and I'm ever grateful for that. And so that led me to UC Irvine. And so at UCI, I was part of the minority science program. And so I joined the lab of Dr. Luis Mota Bravo, where I conducted antibiotic research for 2 years. And so after those 2 years, I took an immunology class, which really sparked my interest into what immunology was. And so that I automatically wanted to do immunology research. And so that led me to the lab of Dr. Albert Zotnik, where I investigated T-cell responses in a multiple sclerosis autoimmune disease mouse model. And so in the minority science program, I also had the opportunity to present my research at various national conferences. And so there I connected with a lot of different scientists from all over the world. And it just opened my eyes to the vast reality of how broad and diverse science could be. So at UCI, I also had the opportunity to become an NIH fellow at the University of Madrid. And so here I joined the lab of Dr. Margarita Del Val. And so there at the Center for Molecular Biology, I conducted vaccine development research. And so this was a really pivotal time of my career point. And so this is the research experience that really solidified my passion for immunology and what impact that could have in human health. So all of these research experiences really confirmed my decision to pursue a PhD in biomedical sciences. And so with the help of the Minority Science Program and all of my mentors, I was accepted to the University of North Carolina at Chapel Hill. And so at UNC, I joined the lab of Drs. Ronald Falk and Dominic Shavada. And so there I investigated disease mechanisms and identified biomarkers for autoimmune diseases such as lupus and ANCA vasculitis. And so doing a PhD was honestly one of the toughest challenges in my life. And it was also the most rewarding. And so there were many times, many days or months where experiments would fail, my papers were rejected, and I really questioned, did I really belong in science? And so, you know, I did have imposter syndrome and I was burnt out and I really considered quitting. But that's the moment, I think, when you find your greatest strength and you look within yourself and you tell yourself, do I want to continue? And for me, the answer was absolutely. That's when I really discovered what the meaning of resilience was. And so for me, resilience doesn't mean being fearless or you know, having all of the right answers right away. For me, what it means to be resilient is you know, holding on to why you started these things in the first place and not letting that be louder than your doubts. And so resilience is also asking for help and collaborating with other scientists, which in the end run produces the best science. And so with the help of my mentors and my loving family right here on the right, I was able to successfully defend my thesis and I earned my PhD. And thank you. And so, you know, during your PhD, you have a multiple wide range of things you can do. You can go into industry, academia, government, etc. And so it was a little overwhelming, all of these options that you could go into. But for me, I really wanted to use my skills that I learned in the PhD to contribute to developing therapies for patients. And so that led me to my drug discovery postdoc at Vertex Pharmaceuticals in San Diego. And so here I worked in the molecular and cell and biology group to advance research and development for early drug discovery programs, such as polycystic kidney disease, which was mentioned earlier today, and by, I think, Edgar. And so, yeah, so in this drug discovery postdoc, I was immersed in what industry meant. And so I really enjoyed the drug discovery part, but I ultimately wanted to be a little closer to the patient end of the drug discovery platform. And so after my postdoc for 2 years, I did an extensive job search. I got many rejections, but I ultimately found a perfect fit at a really exciting startup called Capstan Therapeutics in San Diego. And so in this company, we develop in vivo CAR T-cell therapies for autoimmune diseases and for cancers. And so my current role as a preclinical scientist is to assess the safety and efficacy of drug products before they go into clinical trials. It felt really impactful because this year we got our very first drug therapeutic into a phase 1 clinical trial. And so, you know, looking back on my journey, there's many lessons that I've learned. And looking at all of your young faces today, I just wanted to share some of the mistakes and some of the lessons that I've learned. So first of all, try new things, say yes to new experiences, internships, research courses, side projects, you name it. You don't know what you love until you try it. Another really important thing is to build your network, connect with people in and outside of your field. A lot of these conversations can lead you to unexpected opportunities. Another thing is to find great mentors. I think you're already in great hands with Dr. B here. Seek people who inspire you and to help you grow and find your path. Another big topic for today is to follow your curiosity. Let your passion guide you. The right path may not be linear, and that's totally okay. And especially today in this world, in our climate, your perspective matters, and diversity drives better science. And so your background, your culture, and life experiences bring fresh ideas that the field needs, and never forget that. And most of all, resilience is essential. Experiments will fail, your papers will get rejected, results don't always make sense. But what do? You learn, you adapt, you keep moving. That's how the best discoveries happen. And so 13 years ago, when I stood in this very room presenting my summer research at LA HIP, I didn't know where my journey would lead me. And you don't have to know either at this moment. It may seem like you do, but you don't. So my advice is just to follow your curiosity and just take the next step. And so, especially when you have setbacks and resilience, that's only setting you up for the best science. So thank you so much. This was an honor to be here today, and I can't wait to see where your journeys take you.
Thanks, Carolina. Actually, I have to say, while I was preparing for today, I found on my computer a video, the video of the class of 2012 that you guys did behind my back. Okay, I didn't know it, and you showed it at the introduction to the Science Symposium, and I was tempted to bring that video today, but I thought it's better not. So, anyways. Thank you, Carolina, again. I want to make a comment to your last point on resilience, but I'll do that at the end. Now I'd like to ask Tara Moore to come down here. With this, she will be the moderator for our last session on gene expression and biosynthesis. Tara is also a incoming junior at UCLA. She has been in the program last year. She continued academic year research, and she was in the program this year. She also applied in early 2025 to the Step Up undergrad research program from NIH that I used to have for 16, 17 years. She got accepted. She asked me what she should do, and I said, decline it, and it got cut. The program got eliminated in May, and she would have been in limbo. So thanks, Tara, for this, and I was very happy to have you in the program.
Thank you, Dr. B. Well, good morning, everyone. My name is Tara Moore, and I have the privilege of moderating this section. So I hope you guys are all here with us still, because this next portion on biosynthesis and gene expression is going to be very, very interesting. So to kick us off, I'd like to first call up Nena Mobo to the podium, who is a very passionate second year at UCLA, and she's going to be presenting on complement gene expression in macrophages.
Good morning, everyone. My name is Nena Mobo, and this summer I was focused on investigating the role of epithelial cell-derived complement factor B within the mucosal barrier. Okay, so for some background, inflammatory bowel disease is a group of chronic disorders characterized by persistent inflammation of the digestive tract that affects around 3.1 million people in America alone. It also affects over 100,000 children. Current treatments, there's currently no cure, and existing treatments mainly focus on suppressing the immune response. However, there isn't much research that explores repairing the intestinal epithelial, which is the lining of the gut. Many researchers also study IBD, like Dr. Mark Fry here, so you already know he's an inspiration. So the complement system is a group of proteins within the immune system that helps to destroy germs. There are 3 pathways, and this summer I was focused on the alternative pathway and one of the only key proteins involved in amplification of inflammation called factor B. Factor B is primarily produced within the liver, but its role in humans is generally unknown. Some studies have shown that not having enough factor B could develop into worse IBD, but in people who already have IBD, too much factor B can also worsen it. This is a paper that talks about how a mutation in factor B can lead to a higher risk of Crohn's disease, which is a type of IBD. This leads me to my research question. What is the role of complement factor B in gut cells and its effect on gut inflammation, barrier function, and cell death in relation to IBD? We hypothesized that a loss of factor B within the gut will disrupt the barrier integrity and alter the inflammatory responses, which could therefore worsen IBD. In order to determine if complement factor B is produced in the gut and then potentially its role, we used a mouse model where factor B was completely removed from the liver. This is the albumin-Cree mice, because we know the liver is a main producer of factor B, so we would want to see if factor B, in fact, can be produced by the gut. We also used a full body knockout mice where factor B was completely removed from the mouse's entire body. I then extracted tissue from both the small and large intestine in order to do immunofluorescence staining and validate the presence of factor B within the gut. So a Western blot is a technique used to measure protein levels, and this Western blot shows the baseline plasma levels of the factor B knockout and liver deficient mice, as well as the wild type mice, which basically had nothing done to them. So this confirms that our mouse model works the way that it's supposed to, because you can see no circulating factor B within the liver of the liver deficient mice and the full body knockout mice, but there is factor B present within the wild type mice. I then stained these mouse tissue for factor B, for factor B here, the cell nuclei in DAPI, and cell junctions in E-cadherin. So these images show that there was virtually no effect of factor B on the liver deficient mice within the gut and the wild type control mice, while there was relatively no factor B within the knockout mice. So this suggests that there are in fact gut cells that can produce factor B, and we would want to know more about their production and then ultimately their importance. So in order to determine this, we treated mice with DSS for 7 days. DSS is a chemical that you put in the mice's water source so that we can actually give them IBD, colitis, and make them sick. So we would want to make these mice more sick and then look at them to see if there's any differences in factor B from when they have colitis versus when they don't. So there are a couple of indications that the full body knockout mice, the only mice that did not have factor B in the gut, were getting sicker compared to the wild type and liver deficient mice. So one of them was the fact that the full body knockout mice over time lost more weight than the other 2 controls. We were also able to see that the knockout mice had shorter colon lengths as compared to the wild type and liver deficient mice. And there was also an increased concentration of a protein called TNF-alpha in the knockout mice. TNF-alpha is a protein that alerts your body to send immune cells to a problem area. So in the mice without factor B, more of these immune cells were being alerted. This shows that the gut-derived factor B seems sufficient enough to actually protect mice from injury. So I know it's important to note that in all these studies I've talked about so far, they've all been within mice, but at the end of the day, like, we aren't mice, are we, right? So we're humans, and it would be cool if we could see these effects within the human body. Luckily, I was able to do so. I was able to stain 6 samples of normal human patient tissues and 8 samples of IBD patient tissues. I think you can see that there's a difference in intensity of factor B in the IVD humans versus non-humans, but it's actually important to note that there could be other non-intestinal cells secreting factor B in between the epithelium. So our initial findings suggest that gut-driven factor B has a protective effect on intestinal barrier integrity regulation of inflammation during colitis. And the ELISA data shows that there can be some changes within inflammatory markers in the colitis-treated knockout mice as compared to the control mice. So for future works, the lab is currently working on validating these findings through organoid models and potentially investigating further into the relationship of factor B within humans. I would like to give a special thanks to everyone a part of the Chuck Lorre Scholars Foundation, Dr. B and Dr. Manal Taba, and everyone a part of the Gut Lab and Kulkarni Lung Lab. Thank you.
All right, thank you, Nena. I always love seeing her present because you can just feel the excitement. But next, I'm going to introduce Itzel Cruz, who is a very impressive incoming second year at UCSD, focusing on gene expression and senescence in macrophages.
She's way taller than me, so. Okay, so this summer I was in the Covarrubias lab at UCLA under the mentorship of Graciela Torres, and my research involved investigating the role of cyclin D2 in M1 macrophages. So the Covarrubias lab studies the role of macrophages in aging-related and inflammatory diseases, which is important to understand given the crucial role in the immune system that declines in efficiency as we age. So what are macrophages? Macrophages are cells part of our innate immune system. So essentially they're the first line of defense against pathogens, and they're responsible for the initiation, modulation, and resolution of inflammation. So given their different functions, they adopt several different phenotypes, these being the senescent phenotype, which accumulates with aging, the M1 or pro-inflammatory phenotype, and the M2 or anti-inflammatory phenotype. And so for the summer, I specifically studied the mechanisms underlying M1 macrophage polarization. So in our model, we used 2 mouse strains to replicate immune responses that we'd observe in the human body. The wild-type mice served as controls, and the cyclin D2 knockout mice, where the CCND2 gene, a cell cycle regulator, is inactive. So from both models, we harvested bone marrow to isolate monocytes, which are stem cells. These were differentiated into macrophages over 7 days using a macrophage colony stimulating factor. Then they were plated for experiments, and 24 hours post-replating, they were exposed to 100 nanograms of LPS to induce the M1 polarization. And key features of these cells include transient cell cycle arrest, metabolic reprogramming, and the expression of M1 and pro-inflammatory genes. So, cyclin D2 was initially thought to regulate transient cell cycle arrest in M1 macrophages. At least that was the initial hypothesis that the lab had. However, after conducting experiments, they found out that cyclin D2 didn't regulate cell cycle arrest in macrophages. So this raises the question, if it's not serving its normal function, then what is it doing in this M1 phenotype? A novel finding by my mentor showed that CCND2 regulates mitochondrial respiration in other macrophage subsets. So we wanted to assess if the same was true in this M1 phenotype. So to test this, a seahorse assay, which measures cellular metabolism, was performed on wild-type cyclin D2 knockout macrophages treated with LPS for 16 hours. So here, the y-axis measures oxygen consumption rate, or OCR, and the x-axis measures or shows progression of the assay, not the treatment time. And so in the control groups up here, CCND2 loss impairs mitochondrial respiration, which is shown through the decrease in OCR, suggesting that it has a role in this process. So in the LPS groups, mitorespiration, or down here, I should say, mitorespiration is shut down and we can no longer observe any differences. So based on these results, we wanted to assess if we could capture differences in mitochondrial respiration with a loss of cyclin D2 prior to this mitochondrial shutdown. Excuse me. So my question for the summer continued to be, what aspects of M1 polarization does cyclin D2 play a role in? And we hypothesized that cyclin D2 plays a role in the inflammatory program of M1 macrophages via regulation of metabolic reprogramming. So I ran a seahorse assay again at 2 hours, solely focusing on the LPS group. So the wild type LPS group is this blue line right here, and the cyclin D2 knockout LPS group is the green line right here. And so there is a trend of decreased mitorespiration respiration through the modest dip of OCR, which wasn't previously shown due to mitorespiration respiration shutdown. And so the data from this figure is also used to calculate mitochondrial ATP. And so on this axis, the y-axis measures ATP production rate, and the x-axis just shows you the groups that we studied. And so we can observe that with the loss of CCND2 plus LPS also modestly impairs ATP regulation, further implying dysregulation of mitochondrial function or metabolism. And so given that M1 polarization more than likely involves energetically demanding cytokine production, this led us to investigate if inflammatory gene expression is also affected. And so to assess the expression of these genes, we used a technique called qPCR, which measures gene expression by quantifying mRNA levels in cells. With all of these figures, the y-axis measures relative mRNA expression, and once again, the x-axis shows the sample groups, which remain the same. And so beginning with CD38, which is an M1 marker in macrophages, we see that loss of cyclin D2 plus LPS shows a trend of decreased mRNA expression. CXCL10, a chemokine known to induce cell migration, shares a similar trend. When looking at the IL-6 qPCR data, which is a pro-inflammatory cytokine, we see a significant downregulation of mRNA expression in the CCND2 knockout plus LPS group. And we can see the same for this figure right here in IL-10, which is a cytokine known to be expressed in M1 macrophages. So to summarize, in response to LPS treatment, macrophages upregulate cyclin D2, which plays a role in metabolic reprogramming. I also showed that cyclin D2 modulates expression of M1 inflammatory markers, chemokines, and cytokines. And this is important because it provides a new function for cyclin D2 in macrophage biology, that being that it regulates metabolism rather than the cell cycle, and it provides a mechanistic insight into the regulation of inflammatory pathways. And so some future directions include assessing how this non-canonical function of cyclin D2 is regulated, and also imaging mitochondrial morphology to see what the mitochondria looks like when observing differences in mitochondrial respiration. So I would like to thank you all for sitting here and listening to me, and also my lab. They were very wonderful people, lots of laughs with them, and also to the Chuck Lorre Research Scholars Program for giving me the opportunity to meet such wonderful people. Thank you.
Okay, thank you, Itzel, very poised. So we're almost done, almost out to lunch. I know we're all excited. I personally am. But I'll go ahead and reintroduce myself. My name is Tara Moore, and I'm an incoming junior at UCLA working in the Vlashy lab in the radiation oncology department, mainly focusing on gene expression in different cancer subtypes. This summer, I've been focusing mostly on targeting NRF2-regulated serine metabolism in an effort to overcome radioresistance and sensitize these pancreatic cells to radiation therapy. So the importance of pancreatic ductal adenocarcinoma, more easily referred to as PDAC, is that it's one of the most aggressive and lethal cancers, claiming about 61,000, we're seeing about 61,000 new cases in 2025 alone, and claiming about 51,000 lives this year. There's a survival span of about 5 years, but only around 13% actually do make it to this point. So the issue here with PDAC is that it's just so radioresistant, and when one of the most ideal forms of treatment is radiation therapy, this poses a very, very large issue. So the notorious radioresistance of PDAC is attributed to, in very large part, the serine metabolism axis. Now, when looking at systems that are as complex as this one, I find it easiest to work backwards and digest, which is what we'll do together now, starting here with a decreased effect of a radiation dosage due to decreased radiosensitivity, in large part by ROS, which is contributed to by the antioxidant glutathione, or more easily referred to as GSH. Now, GSH is comprised of 3 key amino acids, cysteine, glutamine, and glycine. Today we'll focus on glycine. Glycine only being produced by being synthesized thereafter. SHMT2, a conversion factor, converts serine into glycine. So now we're going to focus on this serine source, which can come from one of 2 places. It can either come from the endogenous source within the cell or an exogenous source outside of the cell. Now, the thing with pancreatic cancer cells and cancer cells in general is that they are extremely, extremely resourceful. So preliminary research that I've done showed that PDAC cells actually value this exogenous source of serine more, and they favor it rather than using their precious glucose to convert into serine and make it themselves, which, as annoying as it is, these damn cancer cells are so smart. And so that really causes an issue, but for me, it just turned my focus onto the exogenous serine source, in which I focused more so on the uptake through the SLC1A5 membrane transporter, and then the usage of serine thereafter by the conversion factor SHMT2 that converts serine into glycine. Now, in other cancer models, I've seen both SLC1A5 and SHMT2 connected by this transcription factor, NRF2, which then pose the question, what is the role of NRF2 in the serine metabolism axis, and what makes it so important in the radioresistance of these PDAC cells? To which I hypothesized that NRF2 regulates the process of serine uptake and utilization, both SLC1A5 and SHMT2 respectively, thereby enhancing cellular radioresistance in response to radiation therapy. So in order to kickstart this research, I looked at some RNA-seq data from the Gene Expression Octopus database, which is just online for anyone to view, and I saw that there was a very large depletion of SHMT2 and SLC1A5 when there is a constitutive knockout of the gene NRF2, which shows me that there is probably some regulation happening here. Then to tie serine into the story, I ran a few quick Western blots, and I saw that with a depletion of serine, there's both an upregulation of NRF2 and SHMT2, now showing me that there's a connection between enzymes very important in the serine metabolism axis, NRF2, and serine levels itself. That then prompted me to design an experiment with my PI and my postdoc, manipulating 3 key factors: radiation, NRF2 presence, and the addition of serine versus the depletion of serine. So try to bear with me here, it's a little complicated, but in order to do so, there's 8 cell flasks plated, 4 of which are plated in a media with serine, and then 4 of which plated in a media that is deprived of serine. Then, of each 4, there's 2 where there's a constitutive CRISPR-Cas9 knockout of the gene NRF2, and then 2 that act as a control for this knockout. Then of the 2, 1 is radiated by a dosage of 8 gray radiation after 48 hours, while 1 acts as a control to this radiation. Then 24 hours after that, at the 72-hour time point, I'll then run a protein extraction and go through a pretty standard procedure Western blot. So seeing what I found from these Western blots was pretty interesting. Now there's a few things that we can look at here just because we manipulated these samples so much. So what we can see in the first 4 sample groups that have not been radiated is that there's a pretty much a lower expression level of SHMT2, whereas there's a overall higher expression level of SHMT2 in cells that have been radiated, which shows us that SHMT2 is very important post-radiation. We can also then look at a pretty common trend, seeing that serine depletion is actually causing an upregulation of SHMT2 compared to its matched control group. But I'm pretty sure we probably all looked over here at this outlier, which seems to not follow any of the trends that I've discussed right now. And what I'm seeing is that with a depletion of serine glycine, a knockout of the gene NRF2, and a radiation dosage of 8 gray, we see a very stark depletion of SHMT2, which, if you haven't understood anything about my presentation up until this point, is a very good sign. Because if we look back at the serine metabolism axis, if SHMT2 is necessary to convert serine into glycine, and then glycine downstream produces antioxidants which protect the cell from radiation, then if we're able to deplete the serine, as we're seeing here, then that means that we'll be able to radiosensitize these cells and we'll optimize the results of radiation therapy. Now, I do want to point out here for SLC1A5, this data is very preliminary, and I'm working on refining it and running a few more biological repeats. But what we do see here is a pretty common trend, like we saw in SHMT2, that with a depletion of serine glycine as compared to its plus serine glycine control, there is an increase in the SLC1A5 expression, which just kind of solidifies this idea that the exogenous source of serine is very, very important to these cells, and they rely on it very heavily. So what we can take away is that, thankfully, pancreatic cancer cells are metabolically adaptable but not invincible. So by removing this exogenous serine source and working to regulate NRF2 in the serine metabolism axis, this may be a very prime target to potential sensitization of pancreatic cancer cells in radiation therapy. So what I'm going to work on in the next few months to come, and hopefully the next year, is repeating these experiments in PANC1 cells, which are a different line of PDAC cells, just to identify whether this targeting of serine biosynthesis is universal across all cell lines and different types of pancreatic cancer, because that will tell me whether my research is patient-specific or if it's effective across all lines. And it will inform personalized treatment strategies and metabolic vulnerabilities. So I just want to give a big thank you to the Comprehensive Cancer Center and everyone in my lab. A big shout out to my postdoc Prince. It's just me and him in there right now. So we're doing a lot of work. And of course, the Chuck Lorre Family Foundation for supporting me through high school and now through college and doing what I love. And of course, Dr. B. and Manal for all your guidance and your help this summer. Thank you. Now, to finish up our symposium, I'm going to call up a very, very prodigious incoming second year, Rachel Moreno, who studies at Mount Holyoke College, and is going to tell you guys a little bit about pheromone gene expression in C. elegans.
Hi everyone, my name is Rachel Moreno, and this summer I had the opportunity to work in the Sternberg lab at Caltech on the project O-acetyltransferase genes mediate volatile sex pheromone biosynthesis in Caenorhabditis nematodes. Only 40% of human genes have some known functions, and 2/3 of this information comes from studying model organisms. One of these model organisms are C. elegans. C. elegans contain the OAC gene family, which is the largest yet least studied class of O-acetyltransferases. O-acetyltransferases are present in prokaryotes like bacteria to eukaryotes like mammals. C. elegans have two sexes, including males and hermaphrodites. Hermaphrodites can self-fertilize through their limited sperm production. Both sexes mature through 4 larval stages, including L1, L2, L3, and L4 before becoming reproductive adults. Chemical communication is vital for C. elegans reproduction. In my research project, I focus on volatile sex pheromones, otherwise referred to as VSPs, which guide long-range mate navigation. VSPs are produced by sperm-depleted hermaphrodites to attract males. However, their biosynthetic pathway is not yet understood. Based on this understanding, it leads us to wonder which OAC enzymes are essential for the biosynthesis of the volatile sex pheromone in nematodes. Where are these enzymes expressed, and in which tissues is the pheromone produced? We hypothesize that the OAC enzyme participates in the biosynthesis of the nematode sex pheromones. The VSPs may be synthesized in the gonad as it is responsible for producing and maturing gametes. Using chemoattraction assays and GC-MS analysis, my mentor identified 4 OACs required for VSP's biosynthesis. Pheromones were extracted from sperm-depleted hermaphrodites and evaluated based on its ability to attract wild-type HIM5 males. Pheromone extract and M-line control were placed on designated spots on the lids of PG plates, while sodium azide was applied to the agar of the plates to immobilize C. elegans. 20 one-day-old adult males were then released from the starting point, and chemotaxis indices were calculated after 30 minutes. OAC 13, OAC 16, OAC 25, and OAC 28 were identified for VSP's biosynthesis. Transcriptional reporters were constructed, driving green fluorescent protein expression from OAC 16, OAC 25, and OAC 28 promoters, enabling visualization with imaging. Technical challenges precluded the generation of a transcriptional reporter for OAC 13.
Images for embryo through L4 stage include a green fluorescent protein expression image, a DIC image that visualizes internal structures, and a merged image. OAC16 and OAC28 were expressed in hermaphrodite seam cells from embryogenesis to adulthood, and not in the gonad like we had originally hypothesized. Seam cells are epidermal stem-like cells arranged in 2 lateral rows along the C. elegans body. They assist in the generation of differentiated cells throughout development, and no differences in expression were detected between hermaphrodites and males, implicating either additional biosynthetic enzymes or sex-specific substrates in VSP production. In contrast, OAC25 expression was restricted to post-embryonic stages from L2 through adult, suggesting stage-specific regulation. The somatic gonad of C. elegans hermaphrodites comprises 8 distinct structural regions. Notably, the uterine seam sustinctum forms a critical physical linkage between the somatic gonad and hypodermal seam cells during the L4 stage. This connection coincides with VSP production, raising the possibility that the somatic gonadal signaling to seam cells mediated through this physical linkage may regulate pheromone synthesis. While VSPs are not produced in the gonad, its integration with seam cells may provide developmental cues to modulate OAC gene activity. Next steps in the lab include integrating the extrachromosomal line and quantifying the expression level change and expression pattern change at different stages and ages of C. elegans. Thank you to Dr. Wan, Dr. Sternberg, and the Sternberg lab members for your mentorship and guidance. I especially want to thank the Chuck Lorre Research Scholars Program for this enriching experience, and to Dr. B and Dr. Manal for your mentorship throughout the program. Thank you.
So this concludes the scientific presentations. A couple of weeks ago, I went to Caltech to visit Rachel in her lab. And I said to her, let's meet at the cafeteria at Caltech. And I got there, and I saw that smile on her face. And I thought, there's something that I love when students really have some sort of enthusiasm for what they do. And she pulls out her cell phone, and she opens some sort of an application, and I had to put the glasses on because I couldn't see it. She showed me a manuscript from the lab, and her name is on as the author in the middle of it. And I thought, this cannot be true. 6-7 weeks in the lab and be an author on the paper. So I asked her, what did you have to do to deserve an author? And she said, I did all the image analysis of the nematodes. And then she said, it was a little bit tedious and repetitive. And I said, well, that's the nature of science, okay? So, but I also have to say, the PI trusted her, that she really is going to do this right. And I think this is what the Chuck Lorre program is all about, that you go somewhere, you're going in a lab, and you're going to learn how to do this right. And not everything works, and that's fine, but you will learn. This is a learning experience, and you gotta have to take it, okay? So, Manal, could I ask you to go and get the certificates? The students get a certificate of completion, and then all the students, you can choose one of those little bags, whatever you want. But I have to tell you, if you drop it, it's broken, okay? So, all right? So give them a hand, and after the ceremony, there is something to eat out there. Hannah. Tara, you can take it. That's fine. Yeah, that's Jason. So thank you all for coming. And I also have to say, students, you did a really great job. We had rehearsals on Tuesday and Wednesdays, and this is a totally different outcome than the rehearsal. That's why we do rehearsal. So thanks again, and I hope to see you all next year. And outside is something to eat. Okay? Thank you.